enabled the governments to take decisions. How much doctors to provision, how many nurses to provision, how many beds to prepare, how much oxygen to procure, what preparedness will be required, how much will be the wave of COVID, how many patients will be there, how many days it will last. So a lot of such activities were there. Not only that, what kind of interventions you require. So one of the interventions which was we did not have vaccines and we were not knowing about the disease was use of mask. And then there were predictions. If you don't use a mask, your chances are 20% of getting a COVID if you are in a vicinity of a COVID patient. If you wear a mask, it drops to just 1%. Okay. So these kind of calculations. Then another intervention was you stay away. Two people should stay away distance of at least 1 meter. In banks, there were circles, like people were forced to stand on the circles which were 1 meter apart. Right. Wherever there was like whenever there used to be queues, like in banks or in government offices, in post offices, you see that this 1 meter circles were put in. So these are all interventions. How do you arrive at those interventions? By doing simulation and modeling. You see, you have the attributes of the disease and pathogens applied in, and then you put in this measure like if two people are 1 meter apart, how much spread will be there? So you say that if you see that the spread drops significantly, then you have this as a public policy. So such things can be done over modeling and simulation. This has gained a significant, say, a boost this in our times, like it was exposed to the entire world. Okay, so coming back to like, this is something that you must have heard multiple times, but I'll just go through it so that makes a presentation complete. Why do we need IT for biology? The thing is that biological systems are complex. We have hundreds of thousands of different variables, and it is not humanly possible to track everything. Typically have a causality kind of a thing, right? So you see that uh, uh, Aedes mosquito bites you, then you will get malaria. So malaria has a typical cycle. So there is a causality. Like the mosquito bite happens, and then there is a gestation period of 3 days. After that, you will be having a fever. And if that fever is not treated with, say, chloroquine or something like that, then it might result into a very, very fatal disease, right? So we know how malaria progresses. We know how TB progresses. We know how even smallpox, chicken pox, how different diseases which have pathogen based. You have a pathogen exposure. Pathogen grows in your body for a few days, and it overwhelms your immune system, and then you have a disease. If the disease is not controlled in time, then it might become fatal. So this progression we all know. Nowadays, these causal things are taking a back. Of the diseases that we are facing, most of the people die of. So there is diabetes, there is B, pressure, heart diseases, dementia kind of diseases, mental diseases. Are there any pathogens involved? Cancer? No, it's your body going rogue. Your body not behaving in a normal way. And why it is happening? Why does diabetes happen? You eat sugar today, will diabetes happen tomorrow? Answer is no. There are many things involved, including the sugar intake. There are other things as well. There is no causality. It's not like if I eat 100 grams of sugar today, tomorrow I will be detected with diabetes. It is not that causal. The causality is cannot be proved directly, or it cannot happen immediately, because now there are multiple variables involved. And whenever these multiple variables involved, they involve themselves in multiple ways, and that makes it highly complex system. You can't just say that if this happens, then that. There is no if then else statement. Multiple things happen, then you might be hit with disease. Okay. Biological systems tend to be very complex. Okay. And intuition alone is not sufficient to fully understand the dynamics of such systems. Okay. And now the question is that we have unprecedented amount of data that is available to us. And can the AI ML become a potential tool, right? Bring the right medicines to the right patients. Can it happen that way? Okay. And like for example, few months back, Lata Mangeshkar Hospital doctors published a paper in AI. The doctors publishing in AI, right? So an AI-based paper which said that if you do ECG, then with ECG we can find out pre-diabetic patients. We can find that if patient can develop diabetes in next few months, 6 months, 3 months time. And this is a very interesting thing, because sometimes you might have to do ECG, and that ECG now tells, ECG happens for some other reason. That ECG tells whether you are susceptible to diabetes, and then you can do lifestyle modifications or do changes or take some medicines. It might be recommended by the doctors. And then you can pay or even not have the diabetes. Okay. So we see that this kind of data, there are different types of data. ECG is one. There are many times you look at inside your body through sonography, different types of imagings, magnetic imagings, and X-rays and other things, and then find out you might find out something that currently is not disturbing, but might later disturb in your life. So there is a lot of data that is there. Recently, Reliance bought a company which does DNA profile. And with the mutations in your DNA, you can figure out whether are you susceptible to certain cancers or certain lifestyle diseases like dementia or BP or diabetes or even hair fall for that matter, right? So such things can be then taken care of or prevented or delayed. Most probably, we might see a rollout of DNA profiling for masses in next couple of years. And the target price they have is 10 to 12,000. So in 10,000 or 12,000, you might know, are you predisposed to certain diseases? So you might see a revolution here. And that data currently is not available. Like we are not aware that such a thing exists. It might exist in few days, few years. And then we'll have yet one more dimension is available for processing and profiling and trying to find out, can we give right medicine to the right patients? Last but not the least is that the field trials, meaning the actual working of a drug, whether it works on particular set of population, typically very expensive, because you have to produce the drug, take it to the people, you have to administer the drug, you have to track those people for multiple months. And there is a chance of litigation if things go really bad. And sometimes, for example, many of these, if you see the vaccines trials for many of these COVID vaccinations, was you had two groups. One was given just distilled water, and another was given a vaccine. And it became unethical. You know that this group has been given distilled water, and you are exposing them to the world where they can potentially die. Is it ethical? a lot of such unethical questions will be raised. And then many a times, many of those companies modified their protocol and gave all the patients the vaccines. So you see that there is always a dilemma of giving a medicine, a new experimental medicine to the people. So again, that is because it has not been tested. Is it ethical to give? So that question is again there. And if you know that it is effective, then not giving the actual patients that medicine and giving them some distilled water or some placebo pills. So is it ethical? So in both ways, there are many ethical dilemmas that come. And these can be avoided if you can do modeling simulation, find out the effect, a priori effect of these medicines. So IT can come handy here. So there are a lot of exciting things are happening now, and I give some examples. Okay, so there is this In Silico Medicine is a company which has discovered a novel immunotherapy drug candidate using AI techniques for cancer. So it would just target the cancerous cells. So NVIDIA GPUs have enabled simulation of a living cell so that can predict or see how so with the simulation of this living cell, meaning it has not only simulated the DNA, RNA, but also how the cell behaves in different ways, how it divides, what kind of chemical processes happen inside that, and so on. And so now if you can expose this cell to different pathogens, different medicines, and see the effect. Then Integrated Biosciences, MIT and Harvard are leveraging artificial intelligence to discover novel molecules and for suppressing the age-related processes, anti-aging drugs. What is aging? Right? You are the same DNA, you have everything same. Why do you age? Aging has several characteristics, right? So your telomeres get shortened, your bioprocess slow down, so on and so forth. And then the cells don't die that fast. Replacement is slower. And so basically, can you reverse that? You make your body chemical processes same as that were at the age of 20 or 18. And if that happens, it be this will you be the same as at the age of 50 or 60 when you were at the age of 18, 20. So a lot of activities are happening, and these molecules are basically able to mimic the chemical processes that existed far before before you aged. Okay. The evolution and control of drug resistance. So what we are seeing is that like you must have heard there are you should take antibiotics the full dose. You should not take 1 or 2 doses till you feel better and then stop. The reason is that if you don't take the full dose, then the bacteria that have been exposed to the chemical antibiotics survive. If they survive, there is a lot of chance that they might learn the way drugs are presented to them, and their progenies be resistant to that drug. The antibiotics may not work, say 5 months, 10 months, 5 years, 10 years down the line. That is what is happening. So, so the thing is, like for example, our immune system learns when we are given a vaccine for COVID or a vaccine for polio, right? Our immune system is exposed. So, for example, the polio vaccine, it's a live virus, but the virus cannot replicate. It has been modified so that it's a live it doesn't have replication capabilities. So when you give polio vaccine, immune system is exposed to the polio vaccine, and then it learns what a polio virus looks like. And then when it is exposed to the polio virus again, it can identify and kill it. Okay. Then if so there are a lot of drugs and drug resistances they are happening. Especially it is true with the bacterial world. Especially it is true for things that require prolonged exposure to the medicines. For example, TB. So what we are seeing in TB requires you to take medicines for 6 to 8 months. And many times there is boring compliance. People get bored with taking medicines and they don't have compliance and so on. What we are seeing is now strains of drug resistance be happening. It's an evolution of that bacteria from drug-fearing bacteria to drug-resistant bacteria. Okay, and that has to be avoided. And for that, you can have simulations, modeling, you can predict the paths, how evolution is happening, and then control that. Okay. And obviously, then you have models of co-evolution of hosts and pathogens. Many hosts and pathogens co-evolve. Pathogens change, the hosts also change with time. Right? So, like we had seen that the COVID virus is a virus which has been around for a long time, but it has evolved over time. COVID virus was like, if you remember, there was something called as bird flu earlier. And then there was this flu that used to happen through pigs. Don't remember the name. But so you see that it changes the host. And the pathogens change, the hosts change, the hosts are modifying, the pathogens are modifying, and they are looking for new hosts. And that's the reason swine flu. So yeah, so bird flu, swine flu, right? And then we had this current COVID, and then there was a scare for monkeypox, right? So you see that birds, pigs, COVID is assumed to be coming from bats, and we have monkeys. So same family virus which causes flu is going from different animals to different animals. How do you now predict that which animals will be the reservoir for the new viruses? And when these viruses will cross over from that species to the human species? Will it spread? Will it not spread? What will be the potential? And so on. So all these things can be modeled. So mathematical modeling can be used to sharpen our understanding of these processes, as we talked about. And we can talk about alternative policies and interventions that can help us take decisions, can help us prepare for responses. And it can also provide a guide for training and scenario development. And obviously, many times it's a give and take relationship. For example, in COVID, we had lockdowns. These lockdowns resulted in livelihood loss of a lot of people. Okay, so the economy is slowed. India had a recession. Indian GDP was down some 7-8%. And so that's a risk. So what kind of risks we can take? We can have a guided risk assessment, what interventions. So everything has a give and take. What we do and what happens, we have a risk assessment on that. And obviously, then it can also be used to predict the future trends. And so, for example, the flu shot that typically is advised to kids now from 0 to 5 years. How that flu shot is determined is that you assume you collect flu viruses that are circulating this year, and then you find out which has the most chance of survival this summer, and which basically which strain of flu will be contagious, which strain of flu will be most disastrous. You have several of such parameters and then decide a mixture of flu viruses that goes into the next flu vaccine. These are all attenuated viruses, they can't reproduce. Then the next flu vaccine, next year's flu vaccine is made by selection of from the previous year's flu viruses that are circulating based on several such criteria. So essentially, the people who decide on the flu vaccine content doing is they are predicting the future trends. That happens based on your modeling and simulation scenarios. The models are not new. They have been, first model probably was used by Bernoulli in his mathematical analysis of smallpox, 1760. It's a huge time back, but then it's a mathematical model. It's done on pen on paper. Now we do it by coding on computers, but essentially the concept remains the same. And what have these models have highlighted is concepts like core population. Okay, so core population are the population of active viruses or bacteria that are producing the progeny which are most deadly, which are contagious, and so on. Also made explicit the concepts such as herd immunity or vaccination policies. So how do you have a herd immunity? What is herd immunity is that entire people may not be vaccinated. Then you have certain number of people vaccinated, then they will break the chain of virus transmission. The probability that a virus keeps on meeting people who are unvaccinated goes down exponentially if you have several such percentage of people who are vaccinated. And then when the virus cannot find a new host, the current host either dies or is isolated by people because you're seeing the symptoms, or that person's immunity develops so that it basically kills the viruses and the person survives. Right? So that builds up the herd immunity. So if there are pockets of people who are not given the vaccines, that is fine. For example, this polio vaccine is not given to many people in the Western countries because they have reached the herd immunity. There are no people there. So there is no polio strain circulating. So there is no need to give polio vaccines. For India, we will have to still give polio vaccines because there are still pockets, not in India, but then there are surrounding countries where the polio virus is still around, and then there are migrations that are happening or movement of people happening from those areas to India and so on. So obviously the modeling use has led to insights on drug resistance, the rate of spread of infection, trends of epidemics, and the effects of treatments and so on. So this is all we had. So if you have any questions, please don't hesitate to ask. Okay. So moving ahead. What are the modeling tools? We have multiple tools to do modeling. There is agent-based modeling. There is a dynamical systems modeling. There is a modeling which can be done using difference equations and differential equations. Causal modeling, where you say that if this happens, then that happens, and that happens, then this happens, and so on. So for example, if there are rains, Then the trees will grow. If the trees will grow, they will capture carbon dioxide. And when there is less carbon dioxide, there are less heating. There is less heating of the oceans. Then there are less clouds, so less rains. This is like causality. When there are less rains, there is less vegetation. Vegetation meaning less carbon dioxide, more carbon dioxide in the atmosphere. So, which will result in more greenhouse gases, trapping of energy, heating, will result in more rain. So there is this cycle. You can do this causal modeling and then figure out what will be the stable scenario. Will the world temperatures increase by 1% or 2%? What you need to do so that temperature remains down? So this is all causal modeling. Okay? Then there are many mathematical modeling techniques, hidden Markov models, neural nets, deep learning, ARIMA, which is a statistical technique, time series analysis. So a lot of such techniques are can be used. There is probabilistic modeling, modeling of stochastic processes, that is probabilistic processes. Random walk models are there. There is percolation theory, there is Monte Carlo model methods. A lot of these probabilistic modeling techniques are also there. So all of these things use a lot of statistical methods. Statistical methods have long been used in medicine. They used to evaluate the role of chance and confounding association. Confounding meaning the say event A affects event B, but there is event C which is affecting both event A and event B. So can you remove the effect of event C so that we exactly know how much effect of A is on B? So confounding association. So use the sources of systematic changing due to the increasingly huge data sets that we are seeing. And that is calling for research, new approaches. How do you basically use statistical approaches for example, the threat of bioterrorism is now huge. Okay, so how do you basically figure out that things that are changing are not due to natural phenomena, but because there is some things that are being added to the existing environment, right? So, okay. Coming to discrete mathematics and CS theory and its relationship with the biological and medical phenomena. So, availability of large and disparate different databases on subjects relating to disease and diseases and natural phenomena and the processes, natural processes, has basically added relevance to the methods of data mining. Okay, so there are many databases for proteins, then databases on chemical reactions, databases on DNA sequences, databases on DNA to RNA to protein, databases on the reactions on which the proteins kind of act or participate in. So there are a huge number of databases that are available, and then you basically use data mining on databases to find out what is happening, and you can do some causal analysis, or you can do some network analysis, to find the most critical genes or to find the most critical reactions. Okay. And discrete maths and CS theory methods can be used for phylogenetic tree reconstruction. Phylogenetic tree reconstruction is required for evolution, the spatiotemporal and network modeling of diseases. Right, so how diseases are spread geographically, how the diseases are spreading on time. So we basically can use computational biology for disease progression. So if you have a particular disease, progress. So a lot of such diseases can be programmed or basically can be visualized, and then you can have interventions where you can stop or reverse that disease. There are many IoT solutions in Internet of Technology. So these IoT devices are basically these small connected devices to the Internet of Things. And so a lot of these devices keep on collecting the data and information. So you must have, if you happen to see any ward of any hospital, you see that there are a lot of probes that are attached to the patient nowadays. Continuous monitoring of oxygen, blood pressure, and several other things. Okay. A lot of people are also fond of monitoring that themselves by using smartwatches. Smartwatches give you so many things like sleeping patterns, to concentration, BP, pulse rate, and whatnot. Okay. So you see that this has led to monitoring and many times diagnosing of a lot of diseases before they happen. Okay. So you must have heard of stories that the Apple Watch itself dials 911. That is the when you are experiencing a heart attack. So 911 happens to be the emergency number in US. So you can program that ways as well. Okay, so this is what current status is, and we are seeing research challenges we are going to talk about, and some challenges that can be solved in your lab environment. The first thing that I'm talking about is the detection and surveillance using streaming data analysis. So what is streaming data is the data that is continuously coming. And because that is continuously coming, you can't store that data, wait for that data to complete and then start processing. Either let it go. Then typically you used to detect trends and sound alarms. So if something is going up and up, then you say that you are detecting a trend, and then you can, if it goes above threshold, then you basically say that there is something that is not good or not happening here. Okay. So typically, that uses a lot of algorithms that are based on theoretical computer science. Summarizing and storing this data is another challenge. Like when you have that enough data to summarize, or what is your boundary? Where do you, because the data is continuously coming, you have to cut that data into pieces. How do you cut that? Okay. So, if you want to do a disease detection, basically an unnatural spread in particular diseases, how do you do that? Right? For example, this may not happen for humans. Humans are very sensitive. They can post on Twitter or Facebook or WhatsApp or something like that, and things might come to notice. But what about crops, right? If there is a disease on crops, so how is it spreading? It is not spreading. How do you know that? Okay, so there are a lot of data that can come in. They say that satellite images are coming in, right? Or there is a trend in the data is coming for how much market produce is coming in the market. Can you detect because it's happening this way? This year's oranges are very small in size because there is this particular virus on the orange trees. And so if timely intervention is done, we can save half the orange crop, if not full. So such kind of interventions can be done, right? And so and how to design such systems so that it can be monitored and visualized. That is the biggest challenge. That one challenge. Another challenge is basically clustering and analysis of large and complex data. So how do you extract patterns from data? This data may not be homogeneous. This might be heterogeneous. You must have seen clustering techniques where entire data is either numerical or entire data is text-based and so on. But what if the data has numericals, images, some kind of text, and it might be time sensitive also. There might be dependencies on data also. So how there's a lot of heterogeneity. How do you have so no proper mathematical optimization can be expressed on such heterogeneous data? So how do you basically define clustering on such? A lot of research going on here. So newer clustering methods, which are based on theoretical computer science, have to be modified for infectious disease applications. One of the other things is that you might see emergence of new diseases like we saw COVID. Can you predict the classification of this so that you can start giving medicines? So is the COVID virus similar to say malaria? Can we give hydroxychloroquine? It was proposed initially, right? Then it was taken back. So you see that if you do a classification based on the virus characteristic, which is closely related, which group it is closely related to, you might propose some initial things and try it out. So challenge number 3 is basically how do you get a good and clean data in large quantity? There is a lot of dirty data. What is meant by dirty data is that it has inaccuracies. It may have duplications. It may have values missing. It may have values which are so if it is machine reading, and if you read one column, if you column headers change or your rows change, you might have data that is dirty. You might have data. If when you process it, it might give you some very, very wrong results than it should give. If you based on wrong results, then obviously the conclusions will also be wrong. The data is dirty due to various reasons, and moreover, there is no uniform standard for contents and format. If you have standard, then you can compare apple with apple and oranges with oranges. There are no standards. So how do you know that this data is good and this is not good? So for example, uh, if you have a socket, you can fit a Wipro light there, you can fit a Philips light there, you can fit any type of, you can purchase a light, don't care about the brand, just fit it, right? Because there is a standard. That standard decides what should be the size of the the back end of that light that should go inside the socket, right? So in the sense that when that standard is there, then you can compare the lights based on what characteristics you want. One characteristic can be price, another can be say the duration until that it lasts. Third is the brightness, luminescence, and so on. So then you can compare. But you don't need to worry about whether if I buy uh, a luminous light, whether it will fit in this socket or not, because that is standardized. Then in medicine, the standards are missing, or even if they are there, they are not enforced. So there are no data collection standards or something like that, and because of which uh, it becomes difficult to collect the data in the proper format. Okay. So theoretical computer science methods can be used for data cleaning. You can find the data elements that are beyond the common sense things. Yeah. Person's temperature cannot be 100 degrees Celsius. Okay, sometimes instead of Fahrenheit, it might be Celsius. So someone has entered the data but forgot to move that, or basically did not do so much importance whether it was Fahrenheit or Celsius. He just wrote 100. 100 degrees Fahrenheit is perfectly fine. It's a mild fever. 100 degrees Celsius, it's not possible. So, so there might be some such errors. There might be duplicates. There might be required to do merge and purge because a lot of data has been collected which is wrong or having gaps, is duplicates. Moreover, can you do an automated detection of these kind of errors? So this is number 3. Coming to challenge number 4, it's about natural language processing and chatbots. So can you devise effective methods for translation of natural language input to formats that are suitable for analysis? Can you develop computational efficient methods so that you can have automatically answer some follow-up questions? You might have noticed that it might happen with you on multiple times. You went to a doctor, you said something, doctor said something, and then after coming home, you remembered that ye to puchna bhuli gaya. I forgot to ask these questions. I want. Then the doctor is untraceable or is busy or his attendants don't know what exactly is the thing, and you are always like this. The back of the mind, you feel that I should have asked this question. I should have asked whether this is right, this is wrong, or should it be taken with water? This medicine should be taken before meals or after meals or something like that. So this was missing. And these follow-up questions can be answered by chatbot. A lot of data is available. So can such responses be given by scoring the internet, scourging the internet, and then coming up with the follow-up question answers? So that is another thing. So also the data is continuously changing. New drugs are being introduced. New way of doing things or handling of things is being introduced. Can you develop automatic systems, update those queries, right? So you see that medicine is an ever-changing field. Even say a simple thing like cataract surgery. Earlier, you needed to remove and fix a glass-based thing inside the cornea. So you have to remove that disc, opaque disc, and fit a glass. And the glass was moved to plastic. Then it became a silicon jelly. And then basically the silicon jelly can be rolled with us as a nanotube and just expanded inside your cornea so that a small incision is required. Now, this silicon can be filled directly in the retina by making a very, very micro thing so that the patient can go home in just half an hour after the surgery and doesn't require a regimen of 1 month of drops to heal that and like putting up dark glasses and putting medicine in the eyes for 1 month. So a lot of changes happen. This I have seen in a period of 20 years that the cataract surgery has gone from manual thing people used to do. It was a lot of risky thing, and like 10% of the people, 5 to 10% of the people, because of the complexity of the surgery or complexity of their body or non-compliance to the regimen, used to lose their eyesight. Now you see that it is very safe, and it's like going to check your number with the doctor and coming up with the new specs. It has become that safe and simple and easy. Obviously, the complexity has now moved to other things, like how do you develop and design that silicon gel? How do you make the incision? So a lot of robotics and computers are involved. But for the patient, it has become very, very, very simple. So what you see is that now when I query about that, is, you not say you should put the medicines for 1 month and you have 10 times a day put glasses, because now the things have changed. You should have the modern, recent kind of things available, depending on what the procedure has been done on the patient. So, so this kind of things are there. Challenge number 5, containing the contagious disease spread. This is exactly what we had seen in COVID times, like the interventions like shutting down of entire districts or city or even country, and there were containment zones, and there was mask mandate. There were several things that reduced spread of the COVID and that were mandated, keeping a distance of one meter, and reduction in crowded spaces, and on the bus you need to seat only one per seat. In flights, could purchase 2 seats, one and one adjacent seat. So all those things. So typically the diseases are spread through contact, and contact information typically is the key for controlling an epidemic. And so data management tools can be used, right? And data science tools can be used. We can convert a social network as a graph, and we can do analysis of this network, but this network is not static. It is dynamically changing. It's changing with time. It's changing with geography. So you need to make sure that you have strategies in place which will contain this. This is not only for this, like for example, a mad cow disease in US or bird flu that we see, and then you must have heard that so many chickens were killed in such and such district, or the collector ordered all bird fowls to be killed in such and such district. So we see that basically to contain the contagious disease spread. Right? Now there are challenge number 6, that problems that are involved pure theoretical computer science. Like if you are building a phylogenetic tree, so this is an example of a phylogenetic tree. So you have common vertebrate ancestors. You had broad vertebrate ancestors, fishes like that, and then you had tetrapod ancestors. Then you have, so you can see a tree. So finally, so how do you build such? Given the DNA sequences of each of them, will the distance between the DNA sequences, the edit distance between them, or Hamming distance between them, will they form a nice method for this tree, forming the trees? To define an optimal tree. So there are multiple methods: neighbor joining, maximum parsimony. There are many hybrid methods as well. How do you find a consensus phylogenetic? If there are multiple ways, what is the consensus? What is common between all of them? So how do you tailor these phylogenetic methods for viral evolution? Viral evolution is very, very different. You have to go beyond a binary tree. If you see that this is a binary tree, small number of species and large number of internal nodes. But here you might have thousands of vertices which might have very, very high degree. This might happen with viruses. One virus and giving n number of progenies. Not only that, the internal vertex is a live virus as well. So how do you make sure that you can represent this? And moreover, multiple times you might have a virus can be which whose DNA was not produced from one parent, but whose DNA was produced from multiple parents. So multiple parents give rise to the same DNA sequence or same RNA sequence in terms of viruses. So how do you then represent it as a network? Okay. There can be recombinations that have taken place. Two viruses of different parentage combined and formed a new offspring progeny. So viruses are quasi-species in the sense that there is internal core of viruses which is keep on producing different things, but then there is a lot of external core where even if a small change in the virus results in not being viable for reproduction. How do you basically model such virus? Virus modeling is and spread is very, very difficult. Last challenge is basically decision making and policy analysis. Here, how do you find a common pattern in library of, so there is a library of molecular sequences. There are several molecular sequences. DNA is a molecule, RNA is a molecule, protein is a molecule. So how do you find common patterns in that? Okay. Sequence of DNA replicate and what sequences cannot. So like another question is consensus phylogeny in alternative phylogeny. How do you formalize the utilities and costs benefits? Cost versus benefits. How do you formalize the uncertainty and risk? So through a model which has the components of uncertainty, which has the components of risk. Can you define optimization problems for solving like maximum utility of a particular medicine? How do you minimize the pain of a particular patient? How do you design a policy for effective immunization on a large scale? If you have seen the COVID vaccination strategies, are modified with time so that first it was rolled out, then how do you basically keep that 1 meter distance between patients or patients and the people who are giving medicines, who are enrolling the people and making sure that do you see the design of those kiosks, how those places where the COVID vaccination was given? So that design changed over time with experience. Okay. So when you are doing things on a large scale, you have to make sure that all the policies or things that you had given, like there should be mass mandate, 1 meter distance between people, and minimum time spent with other people around. So how do you make sure that all such things are followed even during large scale gatherings, so on? Okay, so these were the 7 challenges I had mentioned. I stop, come to an end my things. I stop with the saying by Hyung Dee, which has been in Nai Chiang, which is a 2600 BC text, a medical text, Chinese medical text. It says that superior doctors prevent the disease. Mediocre doctors treat the disease before it's being evident. Inferior doctors treat the flu and blood disease. Thank you. And for questions, the reference for this is DIMAX Special, so which is basically a computer science society uh, focusing on computational and mathematical diseases. Thank you all for listening, and I open for questions.
Thank you, sir. I request participants, if you have any questions, you can put it in chat box. There is one question, sir. This is just appreciation. That thanking you for this wonderful presentation.
Thanks, Om Prakash. Yeah. Thank you, sir. Thank you.
Okay, I guess there are no more questions. We can conclude here. Thank you, sir, for your enlightening presentation. You gave us the importance of data and IT in biology, some insights about drug resistance, bacteria, how vaccination functions, modeling tools, different challenges such as detection of streaming data analysis, challenges in data cleaning, and the contiguous disease spread, and importance of this theoretical computer science. So presentation has undoubtedly sparked our interest in the field of applied data science in healthcare even further. It has also gave us the deep understanding of current landscape and potential opportunities for further exploration in this research field. Once again, sir, thank you for sharing your expertise and delivering such an inspiring talk. And I hope to have opportunity to learn from you again in the future. Thank you very much, sir.
So thank you from my side as well. It was very good to hear you after a long time, sir.
Yeah, thank you very much. Thanks a lot. So, the only thing I would like to mention before we leave is basically this is an interdisciplinary field which has a lot of ethics and morals because we are dealing with live people. And moreover, if you see the doctors don't see this as their priority thing, doing research or finding out new things. Priority is basically treating patients. So working with doctors, you have to work in with their schedule when they are free, and this you can't this field you can't do research on your own. You have to form collaboration with practicing doctors, and doctors are very busy, and so there is like a doubling of the challenge. Not only you have to find new things for research, but also you have to schedule according to the timings of very busy persons, and they are busy because they are saving lives. They are actually bringing happiness to the people who are under stress and diseased. So at times it becomes overwhelming and difficult to work in this area. But this is the area full of challenges, full of opportunities now because a lot of data is being available. And if you solve even a small thing, it gives immense satisfaction because you affect a large section of people and elevate their diseases, or you can make the drugs more effective, a procedure simplified, and that gives an immense pleasure. So I will encourage all of you to work in this area and do something which you yourself will feel proud of. So thank you all. Thank you there for thank you for listening to me. And if there are any questions, any doubts, you can always mail me, and I will be happy to respond to you. Thank you very much.
Thank you, sir. So participants, conclude this session here, and we will join again at 11:30. Next session will be conducted by Dr. Maitreya Maiti, and the topic in healthcare. So we'll be having 10 minutes short break here. We'll join back again at 11:30. Thank you.